afternoon. As Dr. Slamberg announced, my presentation is about the mechanism of breast cancer progression. Before I start, I would like to share some important facts regarding breast cancer with you. As you all know, the incidence of breast cancer is quite high, and the recent numbers that have been reported is due to the increased awareness and the advances in early detection methods. However, there is about 40,000 women dying of breast cancer each year, and this is only in the US. The majority of these women are dying from metastasis. Some metastatic sites of breast cancer are the lungs and the bones. As you can feel, our main interest is about the spread of these cancerous cells to distant organs, or metastasis. With this slide, I want to show you the process of the metastatic cascade. As you can see, this process consists of several different steps. In the first step, the cells within the primary tumor start detaching from each other, and they start interacting with the extracellular matrix. This interaction with the extracellular matrix activates different signaling pathways, which promotes the release of proteases, degrading the extracellular matrix and allowing these cells to get into the blood vessels. These cells migrate into the blood vessels until they get arrested at a distant site. In here, the release of proteases start again. So this allows these cells to go into the underlying tissue. In here, they start dividing and growing, forming a secondary tumor or metastasis. In order to do our research, we use the breast cancer model, which consists of two different solents, the HS57AT, which will be referred as the parental solent. This solent was obtained from a female breast carcinoma. The other solent is the HS57ATSI, which is derived from the parental solent. This invasive solent was generated in the University College of Dublin by the group of Susan McDonald. What they did was an invasion assay using a microgel, and they found that this invasive solent was 3 times more invasive and 2.5 more migratory, and they were tumorigenic in vivo. When our laboratory finally got these cells, we performed several functional assays in order to see whether we could use these solents. In our first assay, which was the cell-to-cell aggregation assay, we seated our cells on top of an agar gel and allowed them to aggregate for 24 hours. After this time, we observed that the parental cells form a nicely compact aggregate, while the invasive cells form several loose aggregates. So, if we go back to an overview slide, we see that these invasive cells are behaving the same way that the metastatic cells behave, that they detach from the primary tumor and interact as single cells. Our second assay was the cell matrix adhesion assay. This assay was to mimic the adhesion of these cells to the extracellular matrix, in this case, fibronectin and collagen 1. And we can observe by looking at these graphs that the invasive solent adhere 2 times better than the parental cells. And this brings us to our third assay, which is called the wound healing scratch assay. This assay was to observe the migratory activity of these cells. We first measured the width of the wound at time 0 and at time 6. And we observed by looking at this graph that the invasive solents migrate 2 times faster compared to the invasive solent migrates 2 times faster compared to the parental cells. Now that we know that there is some differences between these two solents, we did an staining of the actin cytoskeleton. Why the actin cytoskeleton? Because we know that these play an important role in the movement of the cells. So as you can see, there is a more elongated pattern in the invasive solents compared to the parental solents. Now that we know that this invasive cell line is going through some changes, we want to know what is it that is making these cells behave this well. So, we rely on our three functional assays, but this time using different functional blocking antibodies. In our first assay, which is the cell-to-cell aggregation assay, which uses the antibody against integrin, against coherence. Why coherence? Because coherence is a molecule that mediates the adhesion between the cells. We can see in this image that the parental cells that used to form a compact, nicely aggregate, now is forming several loose aggregates. So, in fact, we see that this parental cell line with the functional blocking antibody against coherence is behaving the same way as the invasive cells. Our second functional assay is the cell-to-matrix adhesion assay. And these cells were treated with different functional antibodies against integrins. Integrins because integrin is the molecule that mediates the adhesion of these cells to the extracellular matrix. And as we can see here, there is not much different in the parental cell line. But if we look at the invasive cell line, we see that the cells treated with the antibody against the integrin alpha-5 beta-1 decrease the adhesion of these cells to the extracellular matrix. In this case, fibronectin. Our third assay, which is the one-heating scratch assay, we treated it with the same functional antibody against integrins. And we didn't see much difference in the parental cell line. But if we look at the invasive cell line, you can see that the cells treated with the antibody against the integrin alpha-5 beta-1 and alpha-V beta-5, their migratory velocity decreased as well. And if you compare it to the parental cell line, it's similar. We know that these cell lines have these more specific molecules, and there are some changes of these molecules inside these cells. So now we want to look how much of these molecules can we observe in the cells and on the cells. In order to do this, we perform flow cytometry here in your left, and you can see that the molecules coherine and integrins slightly decrease in the invasive cell line. And you can see this by looking at the small shift to the left. We also wanted to see the total expression level of these molecules in our cell lines. And as you can see here, by doing Western blotting, these same molecules decrease in the invasive cell line as well. We found, or by looking at previous research, that these two molecules, full-cohesion kinase and SAR kinase, are expressed and are involved in the migration activity of these metastatic cells. So we decided to perform Western blotting to see the activation of these similar molecules in our cell lines. But we didn't see much difference between the two cell lines, as you can see here. But we look at the activated forms, and we observe that the full-cohesion kinase activated at the tyrosine residue 407 is abnormally activated in the invasive cell line. And if we look at the SAR kinase, we also see that the SAR kinase activated at the tyrosine residue 418 is abnormally activated in the invasive cell line as well. We know that in fact, we know that these signal molecules are present in our cells. But we want to see where to localize these molecules. So in fact, we come with some microscopy studies. First of all, as I showed before, is the actin staining. And we can see that in red, the actin staining is more elongated and for a more pronounced pattern in the invasive cells. If we look at the second set of micrographs, we see that the full-cohesion kinase activated at the tyrosine residue 407 is more activated and more present in the invasive cell line at the full-cohesion points where the cells attach to the substrate. What I just showed you was a dual staining, but this time we wanted to see the actin, the full cohesion kinase, and the nuclear staining. As you can see in this overlaid picture, the full cohesion kinase is present at the full cohesion point. However, if you look at the invasive cell line, you see that the full cohesion kinase is more active at the full cohesion sites where the actin fibers end. We not only talk about the activity levels of signaling molecules, but we also refer to the cell surface and total expression levels of some integrins. And one integrin that we talked about was the integrin alpha B beta 5. And we did a study of this specific integrin. Why this integrin? Because we observed that this integrin inhibits the migration activity of the cells. So in fact, in the first set of micrographs, you can see the staining of the integrin alpha B beta 5 and the actin. And if you look at the orange, yellow light colors, this integrin is localized at the full cohesion point where the actin fibers end. If we look at the second set of micrographs, we see that the full cohesion kinase is in green and the same integrin is in red. And we see that the full cohesion kinase is activated at the full cohesion point where the integrin receptors are localized. So you can see here. In conclusion, we prove that our cell model is a really interesting model to study. So, in fact, we observed that the invasive cell line didn't aggregate that well. And why? This can be linked to the cell surface expression levels of cadherins. We also observed that the adhesion of these cells to the extracellular matrix, as well as their migratory activity, can be linked to some molecules, and this one is integrins. This mechanism that when these cells attach to the extracellular matrix activates several signaling pathways as well as signaling molecules. So as you can see here, full cohesion kinase and SAR kinase are activated in this cell line. And by showing some microscopy studies, we were able to observe the localization of these signaling molecules as well. For some future work, we want to go further study the differences between these two cell lines in order to work in human breast cancer tissue. Before I finish, I would like to get this time to thank Dr. Vance Lambrook and Dr. Wim Steelen for giving us this great opportunity of working in your lab. Melissa Candelaria, which she helped us through our research. The School of Science of St. Thomas University, the Science Fellows Programs, and my exceptional lab partners. Thank you. Yes.
Natalia, I have two questions for you. You have a couple of slides with experimental techniques, and you show the relative absorbance over there. Relative to what? And why that relative value is greater than 100%?
Okay, what I just said regarding what I know in my research is that the absorbance of these cell lines were measured at 490 nanometers. That's what I know from my part. This part of this project was also done by different other persons in our lab that maybe they will answer in better words this question. But what I know is what I just told you.
The second question.
Yes.
What is, in your histogram that you show it over there at the bar graph, why some of the molecules you have over there have a different standard deviation errors, that you have the bar errors? In some of them are big, and some of them are practically negligible. What determined that error over there?
Okay, we performed these studies several times, and the percent error that you see showing with the standard deviation is just that between these processes, between the times that this process was performed, we obtain some differences between them each time. But this can be done by different factors that we don't know. But the error that you can see there, as you can see, it's really small. That states that the different performance of our research or our specific assays were pretty accurate.
So why was some molecule has showing more error than others?
Because there were 3 different assays that we performed. So as you can see, they are independent from each other. And there can be different factors that can be affecting these experiments. Yes.
My question was related to Dr. Quesada's first question. Basically, how did you perform the aggregation assays?
The aggregation assays, we first set these cells, the parental cells and the invasive cells, on top of this agar layer. And we allow them to migrate and invade for 24 hours. After this time, we observe at these different cell lengths, and we see that the invasive cell length forms several loose aggregates compared to the parental cell length that was just one compact aggregate. That shows that the molecule that is interfering between these cells, which is cadherins, is not working properly or is changing the behavior in the invasive cell length.
So from your description, I understand how you would get the pictures of the cells, but then how did you quantify them? What was the step now that you get numbers to do, like the absorbance or the quantification?
You're actually mixing up 2 assays for now. The aggregation assay is actually not a quantitative assay. So the only thing you're doing is seeding cells on top of an agar layer. You allow them to form an aggregate. When you're talking about a cell matrix adhesion assay, you're actually seeding cells on top of an extracellular matrix, and then you use a quantification technique, like for example crystal violet staining or alkaline phosphatase, to actually quantify that. And just to answer your question, so indeed, like for example for the cell matrix adhesion assay, the error bars are small. Why is that? Well, because so each experiment is performed 4, 5, 6, 7 times. Each time, per condition, you're using 20 wells, which gives you a certain average. And the more, of course, the more wells that you're using, the smaller your average, your standard deviation will be. And then you compare that as towards the different experiments you're doing. In a wound healing assay, and she showed it shortly, I think you will see it back in all of the presentations, you're measuring a certain width in micrometer. Can start, it depends on how big you make your scratch. It can be 1300 micrometers, it can be 1600 micrometers. Then you allow these cells over a certain amount of time to actually close that gap, and you measure that width again. So there's a lot more variation in that. So that is why the error bars in the migration assay are a lot bigger than in the cell matrix adhesion assay, for example.
Thank you.
So in your wound scratch assay, you use that on the matrix?
No,
No?
it's just
Okay.
on plastic.
Big cells.
In your matrix assays, those are on Matrigel?
No. The cell matrix adhesion assays that we choose to do in this project are collagen 1 and fibronectin. We also, but they were not part of that, we did collagen 4 as well and laminin. Actually, I could use all 4 of them because they all give differences, but with the project of Natalia, we narrowed it down to 2.
More questions? Yes.
How might you relate this to genetics? In other words, looking at the whole thing and taking it to sequence, to genetic sequence.
Our project is nothing related about genetics. Nothing. And I can right now make a link of this assays, a functional assay that were performed to genetics.
So I have a follow-up. So with the Matrigel why aren't you doing it on Matrigel? Is it Matrigel a sarcoma cell line?
The Matrigel was used by the University of Dublin, by the group of Dr. Susan McDonald, in order to obtain the invasive cell line from the parental cell line. They seeded these parental cells on top of a Matrigel gel and allow them to migrate and invade for 90 minutes. And when these gels were collected in the bottom of the matrix gel, they were brought into culture and were called the invasive cells.
I understand that. Why
Yes.
didn't you use that same matrix?
Would I use-
Because they did it. I don't have the intention to repeat what other people have done. Our goal was to obtain a cell model in which we could actually study our research interests. So that's what are exactly the differences between a parental cell line and an invasive cell line, one that forms metastasis. So they had shown that they were 2.5 times more migratory as the parental cell line. So when we got them, and that is what she was trying to say, when we got them in our lab, I mean, those are things that were known. Those people published that. And we just performed a few functional assays that we have running in our lab. Do we see a difference here? If we would not have been able to see a difference, then it was pretty much right over there for me. That is not a model that I can use in my studies. And luckily it was. I mean, it behaves differently.
You're just picking apart the matrix by looking at different components.
Yes.
More questions?